The plant manufactures active pharmaceutical ingredients and pharmaceutical intermediates, an area where the company has many years of experience, as well as the 5-ALA used as raw material for a variety of purposes. A 5-ALA production system based on a strict system of management has been established at Fukuroi Plant. The production of 5-ALA is divided into three main processes: fermentation, refining, and crystallization. In the fermentation stage, photosynthetic bacteria are grown in a large fermenter to produce 5-ALA. In the refining stage, the photosynthetic bacteria are removed from the culture medium, and the 5-ALA and other substances are separated to isolate the solution containing the 5-ALA. In the crystallization stage, the 5-ALA dissolved in water is crystallized, after which it is dried and powdered. So let's take a look at how it is actually refined at Fukuroi Plant. 5-ALA is produced by photosynthetic bacteria. There are special bacteria capable of mass production of 5-ALA. They have undergone gradual improvement and selective breeding as a result of many years of research, without relying on genetic recombination, and play a key role in our production technology. The raw materials used for breeding the bacteria are mainly monosodium glutamate, yeast extract, glucose, vitamins, and minerals, among others. At first, the bacteria are cultured slowly in small flasks. Then they are gradually transferred to bigger fermenters to further cultivate them. Here, the bacteria are being transferred from a flask to a 50-liter fermenter. After that, they are transferred to a 200-liter fermenter, then finally to a huge 35-ton fermenter. The fermenter is 6.2 meters in height. It reaches up to the third floor of the factory. This is a window on the third floor that allows observation of the fermentation state inside the 35-ton fermenter. Incidentally, Fukuroi Plant has an even bigger 115-ton fermenter, and efforts are underway to further boost the production of 5-ALA. After the bacteria have had adequate time to multiply, the raw materials of glycine and glucose needed to produce 5-ALA are fed into the fermenter. These substances are taken up by the photosynthetic bacteria, converted into 5-ALA inside the cells, then discharged into the medium. That's how 5-ALA gradually accumulates inside the medium. That is how fermentation is carried out. Then the next process is refining. The bacteria cultivated inside the fermenter is then centrifuged to separate them from the culture medium. Next, the 5-ALA is temporarily absorbed by an ion exchange resin to remove other substances and impurities. This is how solution containing only 5-ALA is finally extracted. Last is the crystallization process. The purified 5-ALA solution is concentrated inside a clean room, and alcohol is later added to it. The 5-ALA crystals are separated from the alcohol by precipitation, after which they are dried and turned into white powder. These are the main processes by which 5-ALA is produced. Fukuroi Plant is the only place in the world where 5-ALA can be produced with stability using the microbial fermentation method. It will play a central role in providing the world with 5-ALA, for which domestic and international demand is expected to grow in the future.